Right now we're each telling the others what they should be working on but aren't. So anybody have I have a few things listed, but I think I'll hold them on. You guys might bring them up.
Well, I like Steve's comparison of, pop-gen models and macroevolutionary models and looking at how each informed the other and found problems in the others. It's not something we do very often, right? I mean, normally people who do working in macroevolution, you get a tree, you get some traits, boom, bing, bang, you're done. And looking at it from both angles, I think is the way forward for actually understanding this stuff. Not that I'll do that, but.
Yeah, so I think that's something that comes out of a course, out of the course we've got here, and it is conspicuous in the literature as well. You know immediately when you pick up a paper from, say, quantitative genetics versus I don't know what do we
Phylogenies.
The comparative process literature, I guess you might call it. And I think there's latitude for crosstalk.
There is, but as I said in the middle of the week, whether these two areas have successfully made contact is a big issue in this course, and you may come away saying they haven't. You may come away saying, nah, I'm not sure that we can assume that the local change is G over N. Maybe it's something longer term. So it's at least something we want people to come away that we hope we haven't been smug about it.
I think it actually takes some serious effort to cross over between the two and some constant thinking about it, because the methods are so set up in one world or the other that unless you really force the issue, that it's easy to forget which side you're on. But, I really enjoy sometimes just reaching out and pushing the envelope a little bit of my data. So even if I have data on 100 species and I've invented some model to go underneath it, that it's probably not realistic. What happens if you go and estimate a heritability, or like you did a population size or something like that, and then try to take that back? And I agree with you that there's a lot of value to doing that kind of thing and moving back and forth. I'd like to throw in can I throw out a different one that I think is important is space. I've been talking about that quite a bit now, but I think it's a new up and coming area for us to explore, and that we need to do that. There's so much data out there right now that's spatially informed, so geographically informed, and I think we're just starting to. I think one number does not sufficiently describe a species. Right, and so knowing exactly how we want to describe that species, and one standard error on the mean and doesn't do a whole lot either. What I'd like to do is start thinking about how species vary in both space and time, and I think we have some of the resources now that we could start modeling that.
Yeah, I think we've talked about this, but we are at the point where there are methods of collecting a lot of geographic information that it wasn't so easy earlier, and we of course have computers, but there's an awful lot of methods development still left to be done, both at the within species sort of population and quantitative genetic level, and relating that to long-term change in phylogenies. Years ago, I was asked to write, it's a whole story, but I was asked to write for a special issue of Journal of Theoretical Biology, 34 years ago, about important unsolved problems. What was silly was that all but one other author in that volume took a problem they had just solved and declared that it was an important unsolved problem, which I thought was terribly self-serving. Me and one other person put out problems that we didn't know how to solve. But the one I chose was, how can we tell history from geography? If we see differentiation of, say, populations, is it branching history or is it geographic gene flow? People are now there's some good methods. Jody Hay and Rasmus Nielsen's IMA family of programs is one, and there are others in which people are trying to get phylogenies together with gene flow, but there's an awful lot, that story is not finished yet. There's lots left to do there.
So I remember being at a conference on sexual selection. I can't remember how many decades ago, but one of us was trying to be provocative, perhaps me at the end, and I said, like you're saying, but there was a group of theoreticians, and I said, let's discuss the main kinds of models that we need, and the ones that are needed out there, but we don't have right now. And like you say, everybody clammed up because nobody wanted. Everybody's like holding their cards close to their chest like this. I won't show you any of my cards.
If I let you know what I'm about to publish, you'll get it out
So
first.
yeah, it was a sort of embarrassing silence. And I thought, oh yeah, everybody doesn't want to give away their game, but we could do that. We're all comfortable with each other, right?
And the internet.
And the world.
Yeah, of course, at my age, I'm not really afraid of plagiarism, so it's less of an issue. But Amelia and I were talking about whether we could capture sexual selection models with a given AUI framework. And I was saying that I think we're pretty far away because we at least need to be multivariate. You'd really be taking something like its genetic correlations into account. But then there's also this problem that the female preferences are exerting a selection pressure on another trait, the male ornaments.
In another individual.
And it's like that's not even conceived of now, I think, right now in bivariate OU, is it?
Well, I think Hansen has this model where it's OUBM, where you have one trait evolving by a Brownian process and another trait evolving by an OU chasing that.
It's chasing the peak created by him.
Mm-hmm.
And then there's a postdoc here who has work, he hasn't published it yet, but I'm sure it's okay if I talk about it, where it's an OU version of that. So there is that.
Yeah.
MV Slouch has multivariate OU you can do.
Right.
So AUI's not going to do it for you, but there's stuff out there that's coming out soon that can.
So we're trying to. So people are trying to move in that direction. Yeah.
But there's still lots of room to move more towards multivariate processes.
Liam hasn't said anything yet. You have a microphone.
Because your microphone isn't anything.
Yeah. Is my microphone on?
Yeah.
Okay. So I'll say something then. I guess one thing that hasn't been mentioned yet is one of the repeated things that we've discussed is, okay, we want to take into, and this is sort of relates to Amelia's point that you mentioned earlier, which is that we want to take into account interspecific sampling or in the estimation of the mean, but we're basically treating it as if it's a nuisance, but actually that's due to something that's due to intraspecific variability, and that's not something that people are particularly good at studying how it evolves on phylogenies.
I think there was this guy who said that it was involved in evolution. His name was Darwin.
Right.
Yeah.
But the attribute, say, the variance of a population, either the phenotypic variance or its, say, additive genetic variance, studying how that evolves in the tree using comparative methods is not really done.
You mean like the changes of the variance?
Exactly.
Yeah.
And I mean, I don't know why that's the case. I think it'd be quite interesting. I think the problem is I have like a coffee in my other hand, so I'm sort of waving it around.
Talking into his coffee. Yeah.
So the reason for we don't have programs that model the evolution of G on a tree is we don't have an analytical model for the evolution of G. I mean, if you have drift and selection and mutation in there, then which you probably need, then we don't have it. That's why we were doing simulations. You could simulate G on a tree, and that would be an improvement over just doing G and unbranching lineages. So
we certainly don't have the kind of models for changes of something like G where we could calculate the transition probability. What's the net probability of getting from this G to that G? That you have to have in order to do things like a likelihood in a tree. And we almost don't have models at all, and to the extent that we do, we're not going to know that yet.
And so just sort of related to this point, I mean, imagine you have, just in the simplest case, you have the additive genetic variants evolving on the tree. Okay? Well, the phenotype evolving under genetic drift evolves by Brownian motion, essentially, under certain genetic assumptions. Under the same genetic assumptions, the additive genetic variance has an equilibrium value. So it's the process of evolution up the tree is sort of fundamentally different. We don't expect it to evolve, and yet, presumably, G and the additive genetic variants and things like that are different among species. So they're evolving by a process, but under our sort of standard neutral process, genetic drift, we don't expect G to evolve at all unless the mutational matrix is evolving by some other process. Because essentially G is proportional to the mutational matrix under drift. Correct?
Yes.
So
I mean, we could imagine a nonlinear transform situation where when we're in this region down here, a certain amount of mutation makes a certain amount of additive genetic variance. As you move the phenotype up here, now the same amount of mutation makes a different amount of additive genetic variance. So one could imagine some kind of supermodel which predicted different additive genetic variances in different regions of phenotype space.
I actually think that this is something that the empirical work could help with, that we have some foundational work like your stuff showing how G matrices and genetic covariances vary across species, but I don't think we have nearly enough to really inform the modeling that we would like.
But also interesting to think about what happens at speciation, right? So if you have, parapatric speciation versus dumbbell allopatry or things like that, it changes the observed variance within the species, right? But how does that translate to other traits and how they evolve? Mapping that.
Yeah, so I agree with you, Amelia, that it would be great to have a lot more comparative G studies, but I'm fairly pessimistic about, whoops, what does that say? Louder. I'm pessimistic that we're going to get very far. I mean, and that's primarily why I'm resorting to simulations. And the simulation, if you think about what Adam Jones was talking, you guys missed that, I guess, in the early part of the course, but the simulations we're trying to go to more complicated genetic architecture with epistasis. So now we're moving even further away of having an analytical framework for simulating G on a tree. And what we're trying to do is see if epistasis as a component of G, we've already found that, for example, adding epistasis to models enables the mutational process to evolve, right? I mean, I think most of us thought you could hold that as a fluctuating constant, but you add that to the architecture, but you get to some interesting results. You get the result that with epistasis, under pretty general conditions, the mutation matrix evolves into alignment with an adaptive landscape. That makes the G matrix more aligned with the landscape, and now you've got these interesting properties of G that if the landscape changes, then G orients towards it like a little rocket ship, with M2, and so the whole thing is a lot more dynamic. And you can see this all happening in the simulations, but when will we capture that in an analytical framework that can be built into comparative studies? I think it's going to be a long time, I would think, and maybe we're going to have to be satisfied with little approximations. That if you buy into the triple alignment property that we're finding, then you could make little adjustments in G and as you're evolving up a tree, but it wouldn't be. It would be a kind of crude approximation rather than a real analytical thing.
But I'm terrified by that because I have this. I'm terrified by that because if the G's, it's kind of like if the different parts of the universe come into alignment. If the if the G's and the peak movements and everything start aligning a lot, then when we see change in a certain direction, it'll be harder for us to know. Well, we won't. We'll in a sense not be saying it's due to the G being that way or it's due to the selective covariances being that way. We'll just be able to say it's due to all of them being that way. We won't be able to separate as easily the selective stuff from the genetic orientation. I think it's in the cases where the G matrix is oriented off this way and the change is going that way that you get some interesting separation of those phenomena.
I agree. It's more interesting, but I think Mother Nature does not like unaligned matrices. It's and so I think we might end up with that. So I think Schluter's argument, about differentiation being along lines of genetic least resistance, I think that might be a really complicated angle with a lot of things aligned, and you may not in general be able to tease apart the what's causing trajectories if the kind of alignment thing happens that we're seeing.
I think you're arguing for the empirical results there again that you were saying
Am
were
I?
so. Yeah, I think.
No, I think he's demanding that the empirical results fall in line.
Oh yeah. But I think just a few results. Like we wouldn't be thinking so much about. About epistasis or some of these complex phenomena if it hadn't been for just a couple of studies that showed that they're important, right? That over time, and so I think just having a few really well done, careful studies that take you a long time can make a huge difference in the theory, right? Sort of narrow it down.
I want to raise an additional point which I think relates some to this, and that is that I've emphasized, well, there may be limits to our knowledge and we're going to have to find a way to communicate that to the outside world. But in a lot of cases where we, for example, we make inferences about past events that we didn't see, and we try to figure out if it was gene flow or phylogenies, probably the correct answer is not, oh, it's this or oh, it's that, but that there is a collection of possible past hypotheses that we cannot tell the difference between. It's either a tree that looks like this or geography that looks like that or a combination, but not something else. And really knowing not what individual hypothesis is best, but what collection of hypotheses cannot be discriminated, and somehow finding a way to communicate that is probably going to be important, because I see a lot of people now taking a very limited amount of data, 50 species, 6 characters, and wanting to fit models with lots and lots and lots of parameters. And anybody who does that knows that what you get out is not a single point estimate, but you get out ridges through your parameter space, all of which are pretty much going to fit the data to the same extent. And we have to start training ourselves to think that way and to communicate that.
We had a user who once wanted to fit 3 different OU parameters to each branch on a tree with 3 taxa, and is very, very ambitious for the.
Well, but again, if we had a way in the software to say it come back and say, well, you asked me for a point estimate, but I'm going to tell you that there's this whole huge space and all of those fit equally well, that might be helpful for them, in alerting them that they were expecting too much.
We now have warnings like you're trying too hard to get a tri-simpler model. So
Yeah.
it's built in. What do we think about the scale of studies going forward? So now there's something you raised about concerns that over a big tree you have heterogeneity, which is true. But on the other hand, we have methods that can now deal with heterogeneity. Whether it's threshold or hidden Markov models or things like that with a continuous regime, right? So you have more data that way, but you still have even more heterogeneity. So where do we think we should fall on that axis in terms of recommending to people what to do?
Can I comment? I think there's another aspect too of large trees, which is that we have lots of power to reject simple null hypotheses, and that we've had this extensive discussion, and you discussed it I think a little bit as well, about this tendency to sort of conclude that an alternative model that fits better than the null is the generating process for our data, and this is something that we need to make sure that people are not doing. And I mean, understand that if you fit a model, if you fit the Brownian model and you get an estimate of sigma squared, that doesn't necessarily mean that that was the instantaneous variance, the evolutionary process on the tree. That's the best fitting model conditioned on, or the best fitting parameter estimate for Brownian motion conditioned on that model being correct. And I think that this is we've seen it for sort of, we discussed the other day for the Pagal 94 model, also for the state-dependent diversification models. I think the bigger the tree we have, the more likely it is that a simple model is going to be unsuitable for our data. And consequently, the more likely we are to be able to say something interesting with our tree, but in addition, the more likely we are to sort of fit some slightly more complicated model and conclude that model might be the generating process if it affirms our a priori hypothesis that caused us to fit that model in the first place. That's actually probably where the greatest danger lies, is that people are looking for they're fitting a simple model and then an alternative, which the alternative is what they believe it should explain their data. And the bigger tree they have, the more likely that the simple model, say Brownian motion, constant rate diversification, if it's a state-dependent diversification model, is going to be rejected because there's just going to be heterogeneity in the tree, and we have lots of power to reject a null. So that also.
Yeah, I mean, our situation is probably that a null hypothesis that isn't true gets rejected in favor of an alternative that also isn't true. And statistical theory doesn't take that into account too well.
Yeah. But I think that the issue is not that people are developing software to fit models that doesn't work properly. And I think that sometimes people are, but I think how the software and the methods ought to be used isn't being communicated effectively by those that are developing the tools or properly understand these limitations.
So I think that's right. I sense that there's a general feeling in this group that big trees are good. You think that, don't you, Rosanna? Some of them, but they're trees and trees and data sets, but there's some noise in those big trees,
Well,
I guess you were saying.
Yeah, big trees, but they shouldn't be too wide. I mean, a big tree of every conceivable mouse might be much, much better than a big tree of every conceivable vertebrate.
The other issue that arises with large trees is the question of how phylogenetic accuracy or inaccuracy influences the inferences that we make in, say, comparative biology using that phylogeny. And I think, again, we discussed some of the ways in which the effect is somewhat unexpected. I think most of us would expect that it would add random noise. In other words, like, it would cause us to be less able to reject a null hypothesis. I mean, that's what errors tend to do to statistical methods. If you have like a bunch of data and you're trying to compare two groups with a t-test, and you add a bunch of random noise to both groups, then you have less power to reject a null hypothesis if the groups have an equal mean. But in fact, when we do things like add phylogenetic inaccuracy to a tree, sometimes what we actually end up doing is giving ourselves lots of power to reject a Brownian motion incorrectly, because that's exactly what's going to happen. If you have a big tree, you introduce random phylogenetic error, even if it's minor phylogenetic error. It's like the bigger the tree, the more minor the phylogenetic error can be to allow us to do things like reject a null like Brownian motion.
I think also we often fail to recognize that half a tree's branches are the tips. And so people who are building big trees look at, is Ambrella in the right place and stuff like that. But a lot of what's driving your model is stuff happening really at the tips of the branches. And so that's something to worry about too.
To put it in vote for the smaller trees. I mean, I think we've been moving towards these large trees because of the statistical arguments, but the heterogeneity thing is a very compelling argument to me. And I think if the questions that are driving a lot of us, are the evolutionary questions and the wanting to translate this to a microevolutionary level. And I think if there's so much heterogeneity in a big tree, that heterogeneity is going to keep us from being able to infer much biologically that makes sense, right?
I mean, I think I can just think of a study that perhaps you're on, Brian, about, say, woodiness evolving in plants. Is that correct? Yeah. And that's obviously a big scale question, because woodiness doesn't evolve, in a genus for the most parts of plants. It evolves across plant families, and so it's a broad scale question, and the question is very interesting and can be studied precisely, by Brian's approach, precisely because it is heterogeneous across the tree, which is one of the things that they were interested in, I think, correct? So I think that there's a place for it,
but to understand
there's dangers as well.
But to understand the genetics of it, I would definitely go into and look at a few species where it's changing a lot rather than, look across angiosperms. So I didn't think it is question specific what the right level is.
I'm going to present a contrary point of view, in that for a lot of important questions, going across all angiosperms isn't going to get it done either. So angiosperms, am I thinking right? 65 million year old radiation?
No, older than that.
Yeah.
People argue, but let's say
But
140.
a big flowering.
Yeah.
But here's my point. I have really been influenced a lot by reading Simpson's books, which is ironic because my major prof tried to get me to read them as a graduate, and I was going, oh yeah, that's so boring. And but lately I've picked them up, and I started reading them because with Weta and Hansen and a couple other people, we've been analyzing these really deep data sets that I showed you in a couple examples going back, 10 million, 100 million, half a billion years. And on that sort of time scale, it's just not, it's not just an issue of more time for us to see the processes we were familiar with. What I think we're seeing on those really long time scales are things like, little jumps, bursts of evolution that carry you into a new adaptive zone, as Simpsons say, that creates new genera, that creates new families. And we're just, I'm just not going to see that if I study the genus Thamnophis. There are 35 species, I can get them all in there, but there's not an instance of, I'm not capturing the creation of these higher taxonomic groups. And I think that's a whole sphere in which we really need to focus attention and help our paleo colleagues address the origin of higher taxon. I really think we're just not capturing it by fitting OU processes. I'm guessing that we really do need, these Levi process, whatever you want, something that creates these jumps and or helps us, get into, a whole different sphere of integration, however you want to think about it. And that's something we should think about in this course, is how we could pull in a paleo colleague or two. I mean, sometimes we have paleo students. We don't really attract. Does anybody here think of themselves as a paleontologist? We're not really pulling in that sort of.
Yeah, that's actually impressive that there's nobody raised their hand. That's a rather stunning fact.
Yeah. Sometimes there's a postdoc hanging on the edge of the room. At Nessent, we had that a couple times.
So I think, I agree with Steve, and I also think that, for some of these questions, particularly under circumstances in which the paleontological record is not particularly rich, we have no other choice but to depend on approaches like phylogenetic comparative methods. But I also think that, the important thing is to understand how some limitations associated with these approaches become even more significant or of significant concern for large trees and deep time, and so I think that those...
You mean become... I'm not sure whether you're saying that large trees in deep time helps you.
No, it hurts you.
Hurts you, okay.
Yeah. But we have no other, if we're interested in the questions about things that have transpired across sort of broad taxonomic scales in evolution, or that occurred a long time in the past, and we don't have a fossil record that can answer those questions, we either have to throw up our hands or use phylogenetic comparative methods. But if we decide to do the latter, we have to understand the limitations in what they can tell us about these things.
Yeah, well, there will be limitations if you don't have fossil evidence. There also is a matter of integrating fossil evidence in, which hasn't been done enough. There's starting to be some very interesting papers on that, and you have one on how placing a fossil by likelihood in a Brownian motion model, and there's some interesting ones, and people are starting to build in stratigraphic sampling considerations into those models. Now they're in early days, and there'll be a lot more development of that. But you'd hope to have a large tree and then be able to go out and get fossil evidence, tie it into the tree in some reasonable way, that it will then help illuminate those past processes. There are, of course, we've all heard of some particular cases where there are cases where you can get a lineage sampled nearly year by year. These are in relatively recent fossils. If you had a case like that, you can actually, and you could collect enough characters, you can observe the covariances. You can get a very direct estimate of the covariances of evolutionary change without having to deal with deeper branches very much. But ideally, you'd like to bring it all together, and some of the methods are starting to be there.
Yeah, I mean, I think for taxa that fossilize and for characters that fossilize, that kind of record is going to. I mean, it's superior to anything that we can do with phylogenies, but it's just rare.
How about one more consideration? That's genomics. Right now, we're just operating with these characters on scales without any genes. But of course, if you go to quantitative genetics, I got invited to the International Congress of Genetics Applied to Livestock Production, ICGALP, in Vancouver, BC, which is right near, Seattle. It was very interesting because it was the first time in a long time that I've been in a meeting where I would stand around in the reception, and except for 10 people at the meeting, nobody knew who I was. And that's a good corrective to certain kinds of egomania. But in that meeting, what was very noticeable was the classical quantitative genetics that I had been trained in, of variance components and inferring things by breeding designs and so on, was being pushed aside a bit, and everybody was foaming at the mouth about large SNP data sets and using them to map QTLs and things like that. There's a backlash to that coming because in humans, they found too many QTLs of too small an effect. But at the same time, that's going to have to get together with the classical variance component stuff. How is that going to illumine? If we can do that kind of stuff on enough of our tip species, how will that help? Or will it? Or won't it? I mean, we'll be, in a sense, looking at how the characters have changed. We may also get to the point where we can look at the changes of gene frequency at QTLs at the same time.
I was going to touch on that I've been really fascinated by the stuff that's coming out where people are putting aspects of the genome on the trees. So, using phylogenetic comparative methods to study, number of gene families, or some trait like that that's actually a genomic thing.
Yeah. Yeah, I also think it's not a threat to the field, it's a challenge. So in other words, I think that these types of data, we're interested in their evolution, and they evolved on phylogenies. And so, but the problem is, I think if people go about, say, modeling some of these characters using standard methods, they're probably going to find that, that totally different assumptions are required about the evolution of the characters. And so I think that is a future challenge.
Could I suggest at this point that we open up to audience questions like, why are you saying all this nonsense? Or
you can make us fight.
we need to give a microphone to somebody, to Tyler, so he can make it available to people. We do. We have to take responsibility for what we said.
So I have two questions, maybe intersecting. The first one is ecologists over time develop more and more complicated models, and now that they have longer time series and more or better data, more refined data, they are falling back into simpler models, and they notice logistic models very well. Many of the ecological processes, at least for forecasting extinction in abundances. So my question is, where does this field go in those terms? Should we be worried about developing more and more complicated models for these big trees, or putting a lot of effort in like exploring really where the phylogenetic, the likelihood of trees, and get less phylogenetic uncertainty and use simpler models? How you see this going, and where would it be worth it for most of us that are starting our careers to start focusing on efforts towards?
We're each appointing each other to answer the question.
So, well, we got to start somewhere. So I don't think we could go, I mean, so the sort of consensus that builds up, at least I think it's a consensus. I see us a consensus emerging towards the utility of Brownian motion, which is a two-parameter model. I mean, it's not going to get much simpler than that. So I don't think... I don't think there's a one-parameter model out there that's going to do it. But there's a lot of varieties of Brownian motion, and I think although there's a recognized general utility of Brownian motion models, which Joe is extremely happy with, and he should be. But it would be. But I do want to understand which one we're talking about. You know, which variety of Brownian motion. I like Brownian motion of peaks because it makes sort of intuitive sense, and it meshes with a lot of other, studies and quantitative genetics measuring selection and so forth. But that doesn't mean it's true. So I think unlike ecology, I think we're in a better situation, and I come back to Lewitton's phrase that the good thing about population genetics and quantitative genetics is we have these Mendelian roots, which really anchor us in lower level processes like mutation and recombination and so forth. And so they can keep us biologically realistic. And so that's one of the themes I was trying to talk about with the birds of paradise, that we can models of Brownian motion are fine, but we can use cross-links to quantitative genetics to make sure we're, are we assuming a heritability of 20? That would be bad. So we can cross-check, and I think I see that as a way to go, to develop more ways of cross-checking our models and making sure we're in some realistic corner of parameter space, or maybe right dead center.
I would just say that Brownian motion is not, with multiple characters, which is the field has not gone far enough with that yet. Yeah. The OU stuff particularly has not gotten to as many characters as it should. It's a little more complicated than just two parameters, but at the same time, and it's also true that chasing a Brownian moving peak, people have rather glibly said, oh, that'll give you Brownian motion. No, it actually doesn't. If you look at the fine details of the process, it's a little bit different. But our power to examine that is probably very, very small. So it'll almost fit as well, almost fit the same as Brownian motion. I think, though, that we're likely to be in a situation where instead of having a single alternative with two different OU peaks, it'll turn out that a whole bunch of different OU models all fit about the same. And so we may have to either have an easy way of summarizing that, or say, well, the simplest alternative that sort of still fits is this, but there may be more complicated ones that we can't tell from that, even if we reject Brownian motion. So I think some of the acceptance of Brownian motion is just because of lack of power to detect more complex things, and you know, it'd be easy for me to decree that Brownian motion always solves everything, but obviously it doesn't.
I mean, I think it's interesting is people are moving away from, you know, rejecting trivial nulls as the research program and more using models to estimate parameters of things that they care about. I think we're at a good point now where people actually look at confidence in the estimates. That's still coming, and some methods don't do that for you automatically yet. But I think moving towards giving me the best parameter estimates with confidence is the way things are going, rather than OU1 or OU2 or whatever.
But that also relates to the issue that we discussed before, which is that you know, if you fit a, say, model in which you have two rates of speciation and you reject a model which has only one rate, that those two rates are the process under which you know, your data arose. You know, you've actually only rejected a simpler model that was too simple to explain the variability in your data. And so that I think when this movement, which I think for the most part is good, away from, say, just rejecting Brownian motion or just rejecting a constant rate of speciation and extinction on the tree to sort of fitting alternative models, also comes with an associated cost or danger, that these results will be interpreted as you know, the genuine evolutionary history of our group without ambiguity.
Yeah. You don't want to encourage the new person setting out in the field who says, I've got this model that's totally realistic. It's got 270 terms for every conceivable process, and I fit it, and it's better than, you know, naive Brownian motion, because, you know, you want that person to learn that, look, there's probably a whole cloud of processes, and just because you have one that you think is realistic does not mean that that's it. That's what the data is telling you. Are there more questions?
I really wanted to like your comments on the interactions between molecular evolution, molecular process and genomics, and quantitative genetics, because I am like more towards the genomic side, and of course I think like the difference between those two things, I mean, tachogenetics and phylogenetics, are like, to me, are like kind of like Newtonian physics and quantum physics. They're like part of the same or reality. They might interact, but we don't know how, and we have different sets of, well, whatever. So I mean, that sorts of interactions are great, but I'm talking about other like rather trendy or fancy interactions. Like I've seen people doing things like studying gene family evolution as a population genetic problem, or saying that chromosomes are islands and we like can, or transposable element, like transposition is like gene flow between species and stuff like that. So sometimes I don't know at what point can you like extrapolate methods from one side to the other and vice versa, or if that's even possible in a good way. So what is your comments on that? Those like cross talks between fields.
So occasionally you can strike gold just by picking up a model in another field and applying it in a sphere where nobody else has applied it. But I don't know, we should get other opinions, because my feeling about that would be that you'd be damn lucky to do that. I mean, it's probably more likely that a model borrowed from another field isn't, you know, it's like a glass slipper that doesn't quite fit, you know, and it might cut off your toe while, you know, you're trying to get it on. So I'm a little bit skeptical. And you're certainly right about the fad statement, because so over the course of my career, I've seen a lot of things sort of flare up and then die down, you know, and sometimes I could anticipate what was going to die and what wasn't, but not always. So.
I actually think that instead of trying to draw those distant analogies, that sometimes we can just read each other's literature a little more deeply and find things that. So my impression from the genomics literature that I don't read usually is that when I do get in there occasionally, I find something that they've been doing things that are in parallel to us. Sometimes sometimes we're addressing very similar questions, but people are addressing it from different perspectives. And if we would just read each other's literature a little bit more deeply, we would find that solution without having to draw an outrageous analogy. So, I mean, I think there are some comparative methods right now being developed by people in bioinformatics that that we haven't discussed here, you know, and I don't know.
I mean, we could use the idea of false discovery rate more in comparative methods than we do now. That's one thing we take in. I mean, I think it was Barker, Mead, Pagal, one of that set, used just a regular Pagal 94 thing, talked about 2 binary traits to look at predicting gene correlations. We did look like presence and loss across the tree, and found that that method worked better than the traditional approaches that were being used in the field. And so I think in some cases you can bring methods in if you're careful, but other cases you have to be careful of, you know, there's recombination happening within genomes that doesn't happen between oaks and, you know, rushes or something. So keep that things that those in effect, those in thought too.
Yeah, I think that, for example, if you look back in the history of theoretical population genetics, you find R.A. Fisher using physics analogies, you know, talking about when he talked about in the early history of work on diffusion equations, they would talk in terms of heat equations and so on. They were ransacking physics to try to come up with methods, and it worked. It worked pretty well. But then they had to take into account that towards the end of the gene frequency scale, the variance dies away, which it doesn't do with Brownian motion or probably with heat.